Well, welcome everybody. Welcome to the front row. My name is Jamie Williamson. I am the Executive Vice President here at Scripps Research, and we'll be hearing from one of our young colleagues, Mike Bollong, today about regenerative medicine. I'd like to begin by saying a few things about the educational programs we have here at Scripps. We have summer programs where we work with undergraduates and even high school students. We do internships and we host them in laboratories trying to train the next generation of scientists. And we have internship positions during the year as well. But the flagship of our educational program is the Skaggs Graduate School of Chemical and Biological Sciences. So we are a fully accredited PhD program. And we're really proud of this program. We have about 200 graduate students currently enrolled, and we are ranked consistently in the top 10 in both chemistry and biology. And really the next stage of our program is something I'll share just a little bit about, which is the Scripps Research Fellows Program. And there's a reason why I'm bringing this up. Pete is our president, and he decided that we should have a program in place where we capture truly exceptional graduate students right out of their PhD. So typically, once you complete your PhD, you'll go on and do a postdoctoral study at another lab, and then go get a faculty position. And so the idea behind the fellows program is that we would get people right out of graduate school and bring them into Scripps and give them the resources that they need to succeed at a very high level in generating their own independent program without a postdoc. So we set this program up. It's a 3-year program, and fellows are hosted in the laboratory of an established faculty member, and they get salary support and support for some coworkers. And the idea is we give them the opportunity to launch their independent career. So we started this program about 4 or 5 years ago, and we're really proud that the first 3 fellows that we appointed have all matriculated to become part of the faculty. So in the center there's Michael Erb. He gave a front row lecture last year. And then there's Danielle Grochin, is the newest assistant professor to come from the fellows program. She just started this year. And then Mike was actually the very first fellow. He was the inaugural Scripps fellow, and he's also on the faculty, and we're going to hear quite a bit from him today. So we're really happy with this program, and it continues to recruit top scientists. We just had arrive on campus our newest fellow, Shannon Miller, who came from David Liu's laboratory at Harvard. And she's going to be doing gene editing and engineering of things in cells. So we're very excited by the program. It's really been a boon to the livelihood of the Institute to bring in these young scientists and have them thrive. So that brings us to Mike and regenerative medicine. So I think we all understand what regeneration is at some level. Probably most of us, or many of us, have seen, My Octopus Friend, and one of the traumatic scenes is where the octopus gets an arm bitten off, and then it actually can regrow a whole tentacle. So this capacity exists in biology and even in people. So, humans can regrow a significant portion of their liver, but other tissues, not so much. And it's just, there's a very complicated set of signals that any given cell has to start growing or to stop growing at the appropriate time. And so the promise of regenerative medicine is trying to harness the innate capacity of our bodies to repair themselves, but in ways that actually treat diseases. So Mike is going to tell us about this, and his program is a hunt for small molecules and that actually can treat diseases by getting cells to proliferate as if on cue to repair a particular problem. So Mike, it's great to have you on here, and since you are our inaugural fellow and our first assistant professor to matriculate from that program, maybe you could tell us a little bit about how you got started and what brought you to the Scripps program.
Sure, absolutely. Thanks, Jamie. First, I'll say that being a Scripps Fellow was really, truly a fantastic experience. And it, of course, laid the groundwork for the entire laboratory that we have now. I'll admit that it felt pretty daunting at first, the idea of taking control of my own lab straight out of grad school. But, with some excellent faculty mentorship from some of the key faculty at Scripps, as well as being able to sort of leverage the real chemistry and chemical biology firepower of the Institute, I felt like the group took off really quickly. And I think overall, the experiment of the Scripps Fellows program has been tremendously successful with, as you mentioned, the other fellows and the others that you did not. And really, I think the best outcome from my own experience has been in the research program that I was able to create. And the Scripps Fellows program really allowed me to do that. I wanted to work on something that I found that was both incredibly exciting and sort of resembled science fiction, but I also wanted to work on something where I uniquely could best benefit society and medicine. So part of that research program that we crafted together is what I thought I would talk to you folks about today. And that is our work in developing new regenerative medicines and small molecules that can access some of those untapped pathways. So with that, I can get started. So I thought what I would do is kind of break down the talk into roughly 3 parts. So first, I'll give you kind of an intro into our approach and a general idea of what it is we're working on. And the next, I thought I would take you through 2 of the more advanced drug development programs that we have ongoing in the lab that are key collaborations with Caliber, where we're looking to actually develop new medicines. All right, so to start things off, I thought I would tell you a little bit about our research group, given the work we do is a little bit different than some of the work that has been presented so far in this lecture series. So I like to say that our lab is really interested in the earliest phase of small molecule drug discovery. So to do this, we use very large small molecule screens for chemicals to ask whether some drug-like small molecule might exist to answer a very specific biological question that we're asking. So if we can identify a drug like small molecule that does what we want, and that can be anything, whether it be killing a cancer cell or turning one cell type into another, we use chemical tools to figure out how that drug molecule works in cells. And then that's an approach that we often call chemical genetics. And so our research paradigm and what we use to do this has been incredibly useful and ultimately successful to us because it allows us really to do 2 things. One of which is to actually perform academic drug discovery, and that's with the help of Caliber. But it also really allows us an interesting way to sort of cross-pollinate our drug discovery efforts with real just basic science research. And that allows us to do that because we use chemical tools. So it's really in this way that we find new biology that diverges from, but is ultimately complementary to standard biological techniques. So in this way, we kind of really think about ourselves as a genetics laboratory that just happens to use chemical tools to do research. And so that's really our platform, but what are we working on? What are we developing drugs for? And so really one key area that has captured my imagination ever since I started in grad school was regenerative medicine. And I've been supremely fascinated by the idea that you might be able to take a pill and that pill might be able to induce regenerative organ repair. Now that's been a goal of ours and many others for a long time, but regenerative medicine as a field has really failed to deliver on this promise. What is mostly called regenerative medicine nowadays are really only therapies that kind of manipulate the fate of cells. They don't actually promote organ regeneration. So with that kind of in context, our end goal in the lab is to really endow humans with regenerative capacities that other species have. Certainly lower species like newts or axolotls can regenerate their organs. Why can't we? We know this is a hypothetical possibility. After all, we all did come from single cells at one point. Clearly those genetic instructions are there. We just need to learn how to access those genetic instructions appropriately using sort of the tools and the principles of chemistry. All right, so thinking about regeneration, we actually have to think about what regeneration actually means. It means making more functional cells to replace those cells that are lost during periods of damage. So under normal circumstances, the cells in our organs are often making new cells. This process is called cellular proliferation. This is the physical process by which cells make new daughter cells and overall increase the number of cells and the mass of the organ in question. And this process ultimately allows for replenishment and repair of the organ. However, sometimes in the context of diseases and in certain organs and in the context of aging, there really is an insufficient proliferative response in that organ. And due to various processes, these cells die. And there's essentially just a physical gap left over where there are simply fewer cells to execute that organ's function. And so what happens instead is that other types of cells, oftentimes fibroblasts, these cells are ones that secrete scar tissue. This leads to sort of progressive disease, where instead of functional tissue, you get sort of scar tissue deposition and organ malfunction. So that is sort of the key problem. So that was really ultimately the question that we wanted to address. So how can we identify drug-like small molecules that instruct our cells to proliferate and to make more tissue? This would ultimately allow us to make more functional cells and ultimately reverse disease in some cases. So clearly there are a number of caveats to this approach, right? One of which is, how do we do this? What are the pathways that can promote regeneration? How do you turn them on with small molecule drugs? And then of course, lastly, how do you do this safely and selectively? And really addressing that challenge is kind of what I'll tell you about today. So the first of these is our most advanced therapeutic candidate that promotes lung regeneration. And I personally have been thinking about how we might be able to identify drug mechanisms that promote regeneration of the lung for well over a decade. But it really wasn't until about 2013 that we caught a glimpse of how we might be able to actually do this. And that mechanism involves adult stem cell populations. So as you may know, essentially all of our adult organs contain stem cell types that constantly renew these tissues by growing and proliferating. This makes new cells to keep them healthy, and our lung really is no different. So as you may remember from your high school biology classes, where the gas exchanged in your lung are these small sac-like structures called alveoli, and that's shown in the middle here. There are many different cell types in the lung, but for the purposes of today's talk, I thought I would like you to only really remember one cell type, and that's called an alveolar type 2 cell, or an AAC2. So these cells are shown in the alveoli schematic on the right in green, and they secrete a substance called surfactant into the lung, which is essentially a soap-like substance which stops stops the lung from collapsing when you're respiring or breathing. But they also have been shown recently to be the stem cell population of the lower airway. So they both give rise to themselves, so they make new AAC2s, but they also give rise to a cell type called AAC1s, which are in purple. These cells, which are big and are called squamous, they give basically the amount of surface area required for oxygen exchange into your bloodstream. So that's interesting from just first principles, but why are AAC2s important for regeneration? And so it turns out that in addition to keeping the lung healthy during normal periods, AAC2s are really also critically important to a number of disease states. And one in particular is called idiopathic pulmonary fibrosis, or IPF for short. So when I say fibrosis in the rest of the talk, you should think of scar tissue. And given that scar tissue is so salient in IPF, it had really historically been thought of as sort of this enigmatic disease, which really only had been thought of as being relevant to scar tissue deposition, and its origins were not very well known. But what we know now, and this is only within the last few years, is that IPF is really a disease that is driven by insufficient lower airway regeneration. So at the core of this disease and upstream of scar tissue deposition is the ineffectiveness of the stem cell population to grow. So that ineffectiveness of stem cells to regrow and regenerate results in two things, one of which is scar tissue deposition. So scar tissue being in there that decreases organ function, but also different types of cells from the upper airway come in and they proliferate inappropriately. And these two things together sort of result in this runaway progressive disease that is ultimately a death sentence. And really, it was given this information, this new information, this led us to the idea that if you could find a drug that restored that appropriate proliferative response of the AAC2 cell population, you might be able to treat this disease and probably many others at its root cause. So that question, I guess, really hypothesis led us on a hunt to find a drug-like molecule that could do this. So we asked the question, is there some existing drug mechanism that will make the stem cell population grow and repair damage? So what we did was ask the question with 13,000 drugs that have been in phase 1 clinical trials. So we asked the question, is there a drug in the comprehensive drug repurposing library called Reframe that would make these cells grow? So what we did was perform what is called a high content imaging screen with aged human AEC2 stem cells. And what we do here is take many quantitative pictures per treatment to see whether these cells were growing in response to each of those drugs. And at the end of the day, what we ultimately identified was one series of very widely used medications that were the best performers in this experiment. And that was a result that was incredibly surprising. So the drug class that we identified are a set of, as I mentioned, very widely consumed medications, and they are called gliptins, and they're used most prevalently to treat type 2 diabetes. To give you an idea of how broadly used these medications are, sitagliptin, which is marketed as Januvia in the US, and I'm sure some of you have heard of it or know someone on it, had nearly 9 million prescriptions of just this one agent in the US alone. So roughly a few percent of the US population are on these medications at all times. So these are generally safe medications that work by inhibiting an enzyme called DPP4, and I will get into how that works a bit later. But what I first thought I would show you is just the striking results that we noticed when we treated cells in the tissue culture dish with these molecules. This was an anticipated result, but the degree to which this works is really quite striking. So you can see that multiple gliptin drugs, sitagliptin, saxagliptin, and others, expand these cells and make them proliferate or grow in 2 dimensions. And you can see that by the purple staining of the cells in these circles here, which is great. That's a dramatic expansion. But in addition to 2 dimensions, we can see that the drug molecules expand these cells in 3 dimensions as well. This is important because these cells can make what are called alveolospheres, which are like little alveoli in a tissue culture dish, which is thought to really speak to their competency as stem cells. So having both 2D and 3D growth is really important to kind of see what happens and sort of mimic what happens in the adult organism. But as I mentioned, this is the tissue culture dish. And that was really interesting. So we were observing that this small molecule drug class, and many of the drugs in this class did this, essentially all that we tested, and that was in the tissue culture dish. But what happens when we observe this effect in animals? What happens? So this was an isolated phenomenon in tissue culture, but can you actually sort of translate that observation that the cells grow into actually resulting in repair in damage models in animals? And that was a huge leap, but the answer to that question of does this work is yes. So to date, we've evaluated 5 different mouse models of alveolar damage, and the result is incredibly striking. So I don't want to go into the exact details of every single model, but if you just look at the pictures and the damage versus the drug-treated ones, you can see that at high doses, sitagliptin, which is that very widely used medication, results in a dramatic reversal of disease. And seriously, we've never seen anything like it in any of the models that we've done so far. So the effect is really quite striking. So we think in general that we're really tapping into a mechanism that is a central repair pathway in the lung that is used for mammals. So really a key question then was, how does this class of diabetes drugs result in stem cell expansion and work to repair damage? So as I mentioned, this drug class inhibits the activity of an enzyme in your body called DPP4. So DPP4 is a degrading enzyme. It essentially chops up hormones and growth factors and helps regulate their levels in various parts of the body. So in diabetes, it's quite useful to inhibit DPP4 because DPP4 degrades hormones that are involved in controlling your blood sugar. So inhibiting their degradation results in essentially increased levels of those hormones and glycemic control. But what we found is, in addition to those hormones involved in blood sugar control, DPP4 has many other substrates in the body, or substrates or signaling molecules, as you could think about them, that it also degrades or chops up. So what we found after some detective work is that DPP4 degrades certain growth factors in the lung that AEC2s themselves use to grow, which are called IGF-1 and IL-6. So this is what that looks like in practice. AEC2s are at all times making and degrading their own growth factors. And this process is really controlled by the activity of that enzyme DPP4, which is anchored to the cell surface, so to the outside of the cell. And when we inhibit DPP4 with a small molecule drug, we essentially take the brakes off that degradation. And that allows AEC2s to grow and to repair damage, because there is now a stronger signal telling those cells to grow, because there's essentially more growth factor there. So what we are in essence doing is really just augmenting a natural mechanism that these cells use to grow. And this allows cells to repair damage and ultimately to replenish the tissue in the context of these animal models. So now at this point, we figured out how this drug class acts, and we've shown that it works very robustly in animal models. So really a central question is, why can't we go directly into the clinic and start to treat patients with these agents right now? After all, these are approved and largely safe drugs. They should work. And the central reason for this is that there's simply not enough of the currently approved drug that gets into the lung. So the actual luminal compartment of the lung where DPP4 is, for them to work. So it's actually the DPP4 inside the lung which is relevant. So it would require roughly 5 to 20 times the current dose of any approved gliptin to actually achieve high enough drug levels in the lung to observe a likely therapeutic effect in humans. So that was not ideal from a direct repurposing point of view and directly treating patients, but it also gave us the unique opportunity to make a better and safer version of these drugs. Since it's the DPP4 inside the lung we want to inhibit and nowhere else, we decided to make a drug that was directly delivered through the lung to the lung through inhalation. And we modified it chemically such that it would stay in the lung and go essentially nowhere else. And to make it safer and easier on patients, we only wanted to dose it once per week, and we wanted to dose 10 times less than the required oral dose. So that was, in essence, a really tall order, but believe it or not, working with the chemists and the pharmacologists at Caliber, we were able to do that. And so from several hundred potential drug designs that we made, we were able to rationally design our therapeutic candidate, which is called CMR 316. And as you can see on the graph on the left, 316 stays in the lung at very high levels for long periods. So roughly 10,000-fold higher concentration in the lung than what is in the bloodstream at any time over the course of a week. So you can see that it sticks around at relevant concentrations for at least 1 week. And when we look at the body weights or the lungs of these animals, I don't think that you need to be a histological expert to see the difference between the fibrotic lung on the top and the treated lung on the bottom. So as a single agent alone, we think that this molecule will be quite useful in treating disease. But as we start to think about treating IPF, it becomes a little bit different. We also wondered if adding our agent on top of the standard of care, which are the drugs that are used to treat the disease currently, we wondered if those two would add together or together show an effect which is greater than additive. And we think the answer to that question is yes, at least in animal models. So if we co-treat with that standard of care, which is called nintedanib, we observe something which is called pharmacological synergism. And that means that the effect of each of them alone or each of them together is greater than the effect than what would be predicted on each of them alone. So we observe the synergy, we think, because we're essentially adding on a regenerative element to how the standard of care drug works. And the standard of care drug works only really to inhibit scar tissue deposition. It does not actually promote regeneration. So it's this combo of those two things together which results in such a striking phenotype. All right. So based on this, and as you might guess, we are really excited about the clinical potential of this project and this molecule to potentially help patients. And I'm really delighted to announce today that we are very much on track to progress this agent into a first-in-human clinical trial. So last year, we elected 316 as our lead drug candidate, and we're currently performing the required safety studies to take this into a human trial in September of next year of 2023. So hopefully by June of 2024, we should have first-in-human data which reports both on the safety as well as the target engagement of this therapeutic candidate in human patients. All right, so before moving on to the next section, I thought I would kind of recap some of the key points of this project. And the first of which, I think the most important to take away, is that this is, to our knowledge, the only regenerative approach towards treating IPF or any other lung disease. And it really addresses the disease at its root cause. And this is really in direct contrast into how big pharma is sort of developing drugs for IPF, as this is really much more conservative, the approaches that they're taking. We additionally think that these drug molecules will be safe because it really only augments a natural mechanism that the body uses for repair, and that's actually decreased in the context of disease. And it's also worth noting that we're starting with a drug class that has been in many millions of people and has relatively few adverse effects. So we are, in essence, kind of taking a safe drug and make it even safer and better targeted for disease. And I think lastly, it's really worth mentioning sort of the potential broad applicability of this compound series. Everyone always asks me, is this agent going to be useful for COVID? And I think the answer to that question is yes, but it's something that we're looking to address soon. And as you can imagine, it's quite difficult to run the COVID animal models. But beyond COVID, I think an even larger area of impact beyond that and IPF is even potentially COPD. That kills 5% of the world's population annually. So based on our animal models, we've done 2 in COPD. It suggests that we will observe some level of efficacy in this patient population. And just generally, I can tell you that we are really quite optimistic about the future of treating degenerative lung diseases with this agent and others like it. So we think that this will really be a true game changer in changing lung health and how lung disease is treated in the future. All right, so I'll move on to the next set of projects that I thought I would talk about, and that's our work in targeting a key regenerative pathway in animals and its relevance in treating 2 disease states that are associated with insufficient regenerative repair, and those are chronic wounds and heart failure. So previously, I took you through what is called a phenotypic screen. That's where we sort of agnostically look for mechanisms that would promote stem cell expansion. But in this next project, what we are looking for are new mechanisms and really new drug-like molecules that activate a very specific pathway that is involved in regenerative organ repair. So that pathway actually has to do with how our organs control their size. And believe it or not, there is a pathway that controls organ size, and it is shared among all animals all the way down the family tree to flies. And this pathway is called the Hippo-YAP pathway. So just for the sake of simplicity, you can really think about this as two components working in opposition. YAP is really just the gas pedal telling cells to grow until they start to touch each other, at which point Hippo then puts the brakes on cell division once cells are touching each other. It's really this sort of yin-yang relationship between Hippo and YAP that sort of balances organ size on both kind of a micro and macro level. And this pathway was now really, it's been over 20 years roughly since this pathway was discovered. And on the left, it shows what happens when you start to delete the negative regulators of this pathway, the Hippo components. And what you get, as you might imagine, are large organs. So this results in large eyes and heads in flies and very large livers in mouse. So the key question for this talk is like, why does an organ size control pathway, what's its relevance to regeneration? Well, it turns out that YAP is being turned on and off in most of our organs when we're healthy pretty much all the time. And this is actually essential for them to grow and replenish that organ cellular supply. So when we're developing in utero, YAP is very strongly activated, but after birth, it's really only turned on in cycles to control regeneration, especially in response to injury. So this becomes important in the context of diseases where there's an insufficient YAP response. Cells simply just don't grow or grow enough to repair damage. And that observation really led us to the somewhat simplistic idea that if we turned YAP on using a small molecule drug, that we might be able to repair damage and regenerate lost tissue by inducing proliferation or increasing the number of cells in that organ. So based on that kind of simple hypothesis, we really have heavily interrogated this pathway over the last few years, looking for drug-like molecules which might turn YAP on. And so the way that we've done this to date involves screening very large libraries of chemicals for their ability to activate YAP in cells. So you can see that we use the robots that are used in car manufacturing to make this process more efficient. And if we find a drug molecule that works, we validate it in cells to make sure that it's efficacious and selective, and that's idealized by a compound we recently published called PY60. And if we can validate those molecules and molecules like it, then we modify it in such a way chemically that we can figure out what it binds to in cells, what proteins does it bind to in cells, and then we can kind of reconstruct a map of how that molecule binds to a specific protein and how it maps onto that pathway and how it regulates that signaling cascade. And so far, using this approach, we found roughly 6 different drug mechanisms that are unique to our library, or excuse me, to our laboratory for activating YAP, each of which could really be the starting point for a new medicine or a new drug. So far, we've figured out how several of these molecules work in cells, and there do seem to be some commonalities as to how they work. And apologies for the cell biology lesson, but I did want to take you through this just a little bit. So when cells are sparse and they're not touching each other, YAP is free to go into the nucleus and turn on a gene expression program that tells cells to grow. However, once cells touch each other, there are density sensing proteins at the cell surface which tell YAP to be turned off. YAP is then sequestered in the cytoplasm and is inactivated. And what we found is that our drug molecules tend to bind these density sensing proteins and inactivate them directly. They essentially trick the cells into thinking that they are not as dense as, and this results in YAP being active, and that tells cells to grow. And this hopefully results in organ repair in the context of disease. So we now have this arsenal of 6 different mechanisms for activating YAP, and we have multiple ways of sort of progressing these molecules forward. But what diseases can we treat with this pathway? And really sort of a caveat that must be addressed is using these molecules is that you've got to really pick disease states that would be best benefited by a short course of therapy, which would result in healing quickly. Hopefully this effect could also be harnessed within a confined area and wouldn't really require the drug to be everywhere in the body for long periods of time. This would hopefully sort of mitigate potential concerns about inducing or even exacerbating potential cancers that might exist in the body at that time. And so one of those diseases that we first thought of are chronic wounds. So I'm sure many of us have had firsthand experiences with someone who has had a lower extremity ulcer. Just as an example, there are currently 4.1 million diabetic foot ulcers right now in the United States. And if you have a diabetic foot ulcer, there's a 50% chance that within the next 5 years, you will die. So there's really this critically unmet medical need of treating diabetic foot ulcers, because right now, the current standard of care is basically taking weight off of the wound and putting in essentially a fancy Band-Aid to allow that wound to heal more quickly. In contrast, our approach was really pretty simple. It's well known that turning YAP on basically makes the outer and inner layers of the skin grow much more faster. So can we apply a topical drug that would heal the wound faster? It's in treating these types of diabetic foot ulcers or venous leg ulcers, it's really how fast the wound heals that is related to patient outcome and long-term health. So speeding up wound healing really is a huge deal in this context. So as a first step towards addressing this, what we did was see what happens if we rubbed it on the skin of mice that were not wounded, so just normal animals. And the answer was really quite shocking. We found that applying this for roughly a week, we roughly doubled the thickness of the epidermis, which is the outer layer of skin shown in green on the top and purple on the bottom. We also observed an increase in the amount of collagen in the dermal layer, which is the connective tissue in pink shown in the lower panel. And so what this told us was that the compound could really make these cells grow robustly, ultimately sort of reprogramming the normal environment to make skin cells grow rapidly. And that led us to believe that we could make wounds heal faster in people. All right, so it turns out that mouse skin and human skin are actually quite different, and they heal via quite different processes. So a much closer analog to human skin in terms of their composition is the skin from pigs. Pigs and humans have a much thicker epidermis, and our wounds really mostly heal by increasing the number of cells in the epidermis. So our next step was to test our therapeutic agents in non-wounded pigs and see if we could still see this effect. The answer was that we definitely did. We can see we thickened both the outer layer of skin, the epidermis, as well as increase the number of cells in the dermis, which we think will be important for wound quality. We were also able to evaluate this agent in pig models of wound healing. So we were able to make very thick and very thin wounds in the pig. And what we found is that these agents really robustly increased both the rate of contraction, but also the thickness of the wound once it is healed. And that's what's being shown here. So based on the histological pictures on the right, you can see that both the outer and inner layers of the skin are much, much thicker, which would be the pink and purple on the right. And so that gives us really sort of confidence that in the context of a chronic wound, we'd be able to observe faster healing and really observe wounds that are of greater quality, meaning that they would be thicker, more robust, and less likely to occur again. So healing wounds in pigs is great, but pigs, again, as I mentioned, are not really human beings. So the question next is to understand whether this would be useful in human cells and in the context of human wounds. Luckily, there's a way to test this on wound repair on human skin equivalents, which are essentially reconstituted skin cells that have been grown in such a way to mimic human skin, and we can wound them. And what we see in this context is that wound repair occurs much, much faster. It's as fast or faster than the positive control that we were using in this setting, which would lead us to believe we would observe a pretty stellar effect in accelerating human wound closure. Now, that was really interesting, and we thought that we would observe an effect in wounds, but in addition to that, something that we noticed that these agents do, which is very special, is that they make old skin actually resemble young skin. So something that my dad always asks me when I talk to him about my research is, why don't you do something about my skin? So he has a skin condition, which is actually quite prevalent in my family, and it has the unfortunate name of being called Senile Purpura, which means that his skin gets bruised very easily and tears, and roughly 20% of older adults get this quite badly. And the reason why this happens is that because as we age, our epidermis, the outer layer of skin, becomes much, much thinner. But in addition to that, it becomes less attached to the lower layers of the skin as well. So normally the epidermis is connected to the dermis through these projections called reap pegs, which kind of act like a soccer cleat in grass to keep the skin layers from tearing and rubbing apart. In older individuals, there are considerably fewer of these pegs, but there's also less connective tissue or collagen to keep the skin mounted to the lower layers. And this kind of acts like an anchor. So what was pretty fascinating here, and one of the things that we noticed with our YAP activating small molecules, is that the YAP activator essentially corrects all of these defects in aged skin. So one of the benefits of actually working in La Jolla is the closeness to plastic surgeons and our ability to actually get skin samples. So we were able to evaluate for a week on actual human skin from a middle-aged woman who was receiving a tummy tuck procedure, and it was clear what we observed when we treated with the compound. We noticed that epidermal thickness increased considerably, but we also tripled the number of pegs as well as thickened the dermis. So based on this first tells us that we'll likely have some sort of therapeutic effect in live human beings, given that it works on skin taken from a human. But we also thought that this agent might have broad applicability to other disease indications, so for other diseases. So on one hand, we think this could be useful for treating chronic wounds and help save lives, but it may also be a way to rejuvenate older skin that is less mechanically stable. So based on this, we're currently evaluating the best way to move forward with this drug class, and we'll hopefully have some data in higher species soon and decide exactly what we're going to do with these agents. All right, and so that was skin and chronic wounds. And then the last therapeutic area that I thought I'd talk about with trying to intervene with our YAP activating small molecules is heart failure. So as you folks likely all know, heart failure is a huge medical problem in the US and worldwide. Roughly 1% to 2% of the population suffers from this disease, and it costs us over $30 billion as a society to treat. So it's true that we've made tremendous strides in being able to manage heart failure and delay its progression, but ultimately an individual who is diagnosed with heart failure ultimately has a death sentence. And it's really just a question of how long it takes to get there. So really the most common cause of heart failure is a myocardial infarction, which is a heart attack. And when the infarct happens, there's essentially a dead patch tissue that's formed in the heart because blood supply has been depleted there. And when blood supply actually returns, the loss is even exacerbated further. And so overall, this results in a huge decrease in the number of muscle cells, which are called cardiomyocytes in the heart. And the problem is that cardiomyocytes do not regrow after they've been damaged, because the heart is essentially, especially the musculature of the heart, is essentially a non-regenerative organ in humans and most mammals. So what happens instead of regenerative repair is that scar tissue is deposited in the place of cardiomyocytes, and that causes progressive organ malfunction, and that ultimately leads to heart failure and death. And so to treat this disease, what we thought we would do is see if we could tell the muscle cells to regrow with a small molecule that activates YAP, and this should hopefully repair the lost number of cells and actually increase heart function and effectively constitute a cure for heart failure. And so we had thought about this as sort of a very speculative idea some years ago, but really within the last 5 years, the work of Jim Martin and others has shown that this is very much a potential reality. So his laboratory has shown that activating YAP using genetic engineering actually reverses what happens after a myocardial infarction in both mice and pigs. So if you activate YAP using genetics 2 to 3 weeks after the heart attack, you can regrow cardiomyocytes and reverse the decrease in heart function, which is shown on both of these graphs. So now this on its own is a phenomenal result, right? This told us that this was a potential possibility, but genetic engineering of human beings is quite a bit different than it is in the mouse. And I don't know about you, but I would much rather have a non-invasive one-time injection of a small molecule drug rather than taking a gene therapy, which permanently modifies my genes. So based on this, we viewed these data as being very, very promising, but it really prompted us to see if we could recapitulate what had been done with genetic engineering, but instead with a small molecule drug that activates YAP. So we have many of these. So we first asked, what do our small molecule YAP activators do on the muscle cells of the heart, the cardiomyocytes from rats? And so as you can see, the compound induces a very robust expansion of these cells, and they roughly double their numbers every 24 hours that the drug is on them. And what we found is really pretty striking. We found that these drug molecules actually turn on a gene expression program, which makes them both larger and even beat in culture. As you can see in the bottom, these cells are actually beating. So what we think happens in this context is that these drug-like small molecules turn on genes which kind of mimic what happens in both embryonic and neonatal states. So we think we're kind of accessing what happens when the heart is developing in utero when we treat with the compounds. And that works in the context of a living organism to reshape and regenerate the heart in the context of an infarct. So that was in tissue culture. What about living animals? So the first step that we've completed recently is to see what happens when we locally deliver these agents to the hearts of naive adult mice. And what we found is incredibly promising. We found several of these drug candidates increase the number of proliferating cardiomyocytes in these naive animals. So every red cell that is also green is a proliferating cardiomyocyte. And so what this means is that in the context of drug treatment, the cardiomyocytes are effectively reprogrammed and told to grow, and they overcome the very strong the very strong hippo signaling pathway that is activated in the heart, which is repressing these cells, telling them to stop growing. So this is, to our knowledge, the first time that anyone has shown in vivo proliferation of cardiomyocytes with a small molecule drug in an adult animal. So we view this as very promising. We're still at an earlier stage with this drug development program, but as you can tell by how I'm talking, I'm really excited about moving it along. And in the next few months, what we're doing is evaluating if these YAP activating small molecules will show therapeutic benefit in mouse myocardial infarct models. If those mouse experiments are successful, we will demonstrate that effect in pigs and then subsequently perform the required safety studies to take this molecule or one of it like it to the clinic by 2025. So hopefully by 2025, maybe the next time that I give this lecture, hopefully I can give you some good news about this therapeutic effect in human patients. All right, so lastly, I would just kind of like to recap some of the key points of the YAP section. So first, I'd like to emphasize that we found drug-like small molecules that activate YAP, and these really essentially just augment the body's own growth control pathway. It's just that we found ways to reactivate these pathways when they are off in the context of disease. And we think this will really be of critical importance for disease states like the ones I talked about, which are chronic wounds and heart failure, where there is an insufficient growth response and YAP is really turned off. And so lastly, I think just in general, we're really excited about the potential of these regenerative therapies. And I hope that I've conveyed some of that excitement and hope for the future in treating these, what are presently incurable disease states with regenerative therapies with all of you. All right, so I will leave the presentation at that. So first, I'd like to thank the really fantastic people in my lab who did the work. The AEC2 work was really spearheaded by the tremendously clever and talented work of Sita, who was helped considerably by Greg. The YAP work was initially spearheaded by Edita, especially the earlier phases of discovery, and that's been passed to Maya and Kayla, who are continuing that. I'd also like, as I mentioned, to thank the Scripps Research community. This includes some really fantastic faculty mentors, namely Pete Schultz, Luke Wiseman, Luke Larson, and many others. And lastly, I'd like to thank the folks at Caliber. They've taught me a lot about drug development in the last few years, and they've really kind of brought some drug development know-how to make some of the kind of wild ideas that I've cooked up into actual therapeutic candidates, and I'm excited to work with them. And so lastly, I'd just like to thank you all for tuning in and your attention. I'd be delighted to take any questions.
Thanks. Terrific, Mike. There are indeed questions, quite a number of questions. I think that was a great job explaining some pretty complex chemistry. Let me ask a question. Let's start out with your lung regeneration work. So you showed us a lot of histology, and is there a way to actually assay the lung function? You know, can you put the mice on a treadmill and is there a way to directly measure that?
There are. So there are imaging techniques, and there are also ways of measuring FVC's forced vital capacity in larger organisms. But we found that mouse really nicely recapitulates what we need to look at, which is mostly fibrosis and repair. So we've actually shown that. So we haven't gone in and done those actual studies in larger animals, but we have shown that this compound class does promote barrier function. So the actual barrier integrity in the alveolus is very much intact when they're treated with the drugs. So we know that we're getting repaired. And we've also gone in and done something which is called single cell RNA sequencing, which allows us to look at the gene expression profiles of every single cell type in the lung simultaneously over multiple time points. And so what we can see is that indeed, I didn't have time to talk about it today, but what happens is indeed the compound does instruct the stem cell population to sort of take on this transient phenotype, turn into an AAC1, but also maintain its stemness. So we've proven sort of conclusively, at least in the mouse, that we are inducing repair, and that results in increased barrier function.
And so when you do that, after you've repaired, do you have to continue therapy, or is the effect does it persist after you discontinue therapy?
Fantastic question. That is still TBD. We think in the context of IPF that it will be a chronic therapy. So in lung fibrosis, that stem cell population is permanently just non-proliferative. So we think that that's just not working. So if we can turn that back on, we'll likely have to keep putting the gas pedal down for us to observe regeneration. In other disease states, we don't know if that's the case yet. Certainly in the context of a shorter disease like acute respiratory syndrome, which is say you have a viral infection and then you have lung damage, that would be only a short period of time, or for example, COVID. But in chronic disease, we think it will be chronic, but in other cases, it may not be.
So a number of people had this question and cast in a variety of ways. I'll just paraphrase it. So the compounds you're finding cause cells to proliferate, but cancer is a disease of proliferation that's unchecked. So what's the balance between turning cells on and making sure they don't go too far, and do these exacerbate any kind of cancers?
Yeah, that's a key question at the forefront of my mind at all times. And yeah, the way that we are addressing this is multifold. The first of which is really to select diseases where proliferation is missing, right? And then another way that we're addressing this is to deliver these drugs locally and hopefully for short periods of time. So in the context of the heart, for example, we're delivering the drug directly to the site of infarct. It makes the cells grow, it sticks around once, and it's a one-time injection. So we think in that case is a good use case, not going to induce cancer of that population. And that's been shown using genetics as well. Yeah, and then the last thing is sort of how we design these small molecules. The drugs aren't getting everywhere and they're very quickly cleared. So we're delivering them locally. And as soon as they leach out of where we put them, they're very quickly metabolized. So they're either not serum stable or they're metabolized by the liver and quickly cleared by the body's own metabolic processes.
Right. Now, you started out the lung work was done, you talked about the Reframe library, which was the set of all compounds that have ever been in people, which is about 13,000, I seem to remember. And what, so a couple of people were asking about the screening of those, and is this something that you do the whole library against every target? Or are you using computational approaches to kind of pre-select subsets? You could say something about that.
Sure. So I'll first say that using Reframe has been just a tremendous resource for my lab because we're interested in stem cells and finding new pathways and repurposing in general. But typically what we do is actually just screen the whole library. Mm-hmm. As I mentioned in the talk, we often can screen almost 1 million molecules with Caliber. We have a few 100,000 in my own laboratory. So this is a relatively small collection. So if we have a biological experiment that accurately reports on the biology going on, screening 13,000 is certainly doable and one that I would recommend doing because this is basically just sort of multiplying serendipities. And sometimes you find something that's very interesting that can be repurposed quickly and tells you something new about biology.
I mean, 13,000 sounds like a lot, but compared to technology, it's really not. And it remind me that I missed this or just, but the YAP activators, did they also come from the Reframe?
No, those are all novel molecules. And, okay. I wish I had more time to talk about them, but what's so curious about this is that all of the targets that we found for these novel molecules are novel components of the Hippo pathway as well. They're not annotated targets that are known to be as sort of core members. So there's kinases in that pathway, which are enzymes that do phosphorylation. Those are not key targets. We've never found any kinase inhibitors. We've all identified new targets for those. So a treasure trove of information for someone who likes chemical biology like me.
Right. So I noted that the second two parts of the second 2/3 of your talk were all YAP activators, but it was two entirely different tissues. And so how general is that YAP pathway, and how many other such general factors are there that could be where this approach could be used?
Yeah. That answer of are there many general factors is very few. So among regeneration in mammals, YAP is one of the only games in town. So that's why we're hitting it so hard and looking for so many different ways to activate it. We think that there are specific ways to turn YAP on in a given tissue. So maybe like tissue restricted ways of activating YAP with various small molecule drugs. So that's really what we're looking for, is to say, let's take the right YAP activator with the right mechanism and use it for the right disease indication. And hopefully we'll find something that works quite nicely. And I think for the heart, that's one where we found a really superior mechanism that we've not yet disclosed, but we're excited about.
Let's see. So I think actually a couple people asked this question, and it seems like another indication would be, ulcerative colitis or irritable bowel disease. And have you thought about that? Do you have programs in that, or?
Yeah, no, we've actually evaluated some of our molecules in IBD models, and they show sort of modest efficacy. YAP in the context of the gut and the colon isn't as simple as it is in, say, a chronic wound where you just want things to heal as quickly as possible, or in the heart where you just want cardiomyocytes to grow. It seems that YAP is a little bit more subtle, and it seems to be more of a damage phenotype that's activated in IBD, and it's not always beneficial to just regrow the gut, and that doesn't always translate to disease efficacy. So the gut is probably a use case of it not being so useful, but there's probably others that we're not even thinking about that would be benefited by a YAP activator, especially a specific one.
And maybe the last question I would ask you, last science question would be, everything you talked about was developing small molecules, so drugs that provide some therapy. And for regeneration, the other approach is, of course, stem cell therapy. So could you give the 100,000 foot, over under on those two approaches?
Yeah, well, first I'll say there's a reason why I'm working on small molecules and not cell-based therapies, because I think manipulating the fate of endogenous stem cells, or the stem cells that exist in your body, is a much, much more likely way to succeed. And I think that's been shown. Stem cells and their direct transplantation into humans has been, unfortunately, not what we thought it would be, and is not as useful as was predicted. There's some therapeutic efficacy from inducing mesenchymal stem cells into certain populations. Obviously, people get shots of these cells all the time, and their efficacy is questionable, seems to be person to person. But replacement therapies using reprogrammed stem cells, I think that there's a long way to go, and a more direct route and potentially more efficacious route, and one where we can succeed in particular, is to find molecules that change how the stem cells that exist in the body, how can we manipulate them to repair damage? And that's kind of been my mission statement ever since I started.
Yep, no, it's clearly really promising. It's super impressive, and I can assure you the audience has a lot of enthusiasm for everything that you talked about. So thank you for that. So the last question I would ask, this is just a personal question. So Mike, how did you turn into a scientist? Where did you come from, and how did you get here?
That's an easy question. So it was really the fantastic science being done at Scripps, which made me want to become a scientist. So I was actually an undergraduate researcher way back in 2008 or 9, and I was working in Pete Schultz's lab. And at the time, I thought I wanted to go into medicine, but as soon as I saw what was being done using the power of chemistry, I was in awe. We were working at the time to reprogram skin cells into stem cells using small molecules, and that you can see that there's been really a consistent thread in the thinking there, and it's brought together my interest in stem cell biology, regenerative medicine, and drug discovery, and that's just sort of morphed into the program that you've seen today. So I guess you could say I got the bug, and I haven't been able to stop thinking about it or working on it since.
Okay, well, you're early in your journey, and it's looking great. So thank you very much. I just want to make a couple of other just reminders to everybody before we sign off. So we have a podcast that goes on, and it's actually very entertaining. We have episodes almost weekly, and if you're interested, you should tune into this podcast. So we have 3 of them queued up on a variety of interesting subjects. And then lastly, this is the last lecture in the front row for this season. So thanks for the cleanup duty, Mike. But we wanted to just give a preview of some of the upcoming talks, which would be in the fall. And we have 4 really outstanding speakers with a broad array of topics that I think you'll find interesting. So you can get a recording of this front row lecture on the frontrow.scripps.edu website. And actually, we have a transcript of all the questions. There were a lot of questions that we couldn't get to, and Mike will have the opportunity to respond to you if we can arrange that. So listen, it's been a great year. We're glad to finally kind of be out from underneath COVID for the most part. I'm hopeful that we'll actually resume a hybrid meeting in the fall where there'll be people in person and video. I don't know if that's been decided yet, but I'm certainly looking forward to seeing people in person and welcoming people back to campus. So thanks again, Mike. And again, I'm Jamie Williamson, and thanks for coming to the front row. Bye.